Thank you all for spending part of your Thursdays with us. My name is Lauren Snyder, and I am a postdoc at the German National Library of Science and Technology, also known as the TIB. And I work in the lab knowledge infrastructures, which is led by Dr. Marcus Stocker, who is also on the webinar with us today. So on behalf of Marcus and myself, I want to welcome all of you and say thank you so much for being here. We're really excited to have you all and to also introduce this new open source approach to producing scientific knowledge, which we are calling reborn articles. So to start us off, I'll give us just a brief overview of how we've structured this webinar. So I'll start with just a brief introduction of the reborn articles approach and tell you a little bit about our motivation and why we think this is something that's needed and it's important. And then the most exciting part of the webinar will be when I turn it over to my wonderful colleagues, Dr. Ana Maria Mian Marquez and Dr. Alberto Pina. They're both early career marine biologists who have been working with Marcus and I through a curation grant for the better part of half of a year now to implement reborn articles, not only on some of their scientific work, but also from other members of the research community. And they've also given us tremendous feedback in terms of how we can improve this approach to make it even more useful for you as researchers. So it's really thanks to both of them that we're here today and that we have this webinar. Then I'll just quickly jump back in to explain very briefly how this approach works, how we do this, so you better understand how you can get involved and how you can apply it to your own research. And then we will finish with a Q&A discussion. So I want us to start in a happy place. So think back to the last time you were planning a vacation. So I'm thinking about my trip to Bologna, Italy last spring, which technically was a work trip, but it felt like vacation. So let's now have our vacation destinations in mind. And I want you to remember, how did you figure out how you were going to get from the airport or the train station to your hotel? Did you use a physical paper-based map to do this? Or did you use a digital navigation app like Google Maps? My guess is that most of us probably use something like Google Maps, and this is a great example of the effects of digitalization in our everyday lives. So a couple decades ago, we all would have had to use a physical map to plan our route from the airport or the train station to the hotel. Fortunately, this is much easier today because we can just give Google Maps our destination, click Plan My Route, and then we're done. We can even zoom in and look for other interesting places nearby. But if we consider how we interact with scientific information, we see that this advancement from analog to digital is a bit different compared to other aspects of our everyday lives. So when we compare the first scholarly articles that were produced in the 1600s to what we're producing today, it's clear that the underlying mode of how we share this information has not changed. We still rely on text-based articles. So if you think about it, in nearly 400 years, we've managed the switch from physically printed articles to PDFs, but nothing more. So if we come back to our navigation example, it's as if we're living in a world where we never developed navigation apps, but we instead rely on PDF scans of maps. So for science, we now have these PDF articles that can easily be shared electronically and that allow us to easily structure an article's metadata using reference managers. But most scientific data cannot be organized in this way. Why? Because we're not producing them in a format that machines can interpret. So in other words, they are not machine readable. Rather, we bury crucial information in PDF documents, and this limits our ability to use digital support tools that could make the research lifecycle more efficient. So for example, think about how time-consuming it is to manually extract and organize large amounts of information for something like a meta-analysis. And yet, efficiently synthesizing findings from complex data sets is exactly what we need to be able to do to address challenges like climate change and biodiversity loss. But what if we could produce our scientific data in a machine-readable format? Then, as with navigation apps, we could leverage machines to interact with scientific knowledge in entirely new ways. Reborn Articles is a new open source approach that allows us to do exactly this, produce scientific results in a machine-readable format from the outset of production. And we recently created a digital library, ORKG Reborn, specifically designed for Reborn Articles. The approach integrates with programming languages that are commonly used in data analysis, like R and Python, and it ensures that scientific findings are born reusable by humans and by machines. And this is where we get the name Reborn Articles. By producing results in a machine-readable format, we enable ORKG Reborn to automatically collect and organize them, bypassing the need for post-publication data extraction. So today, Ana Maria and Alberto will show you how implementing this Reborn Articles approach on their own published scientific work allowed them to transform their scientific results from a standard PDF format that was not machine-readable into a highly structured machine-readable format that was automatically collected by ORKG Reborn. Now, researchers anywhere in the world can download their data as an Excel file and copy the underlying code into a computational environment like RStudio so they can recreate the results for themselves. So to summarize, by implementing the Reborn Articles approach, machines are now able not only to collect the metadata of Ana Maria and Alberto's articles, but also the main results. So now that we have a basic understanding of how this approach works and why it's important, I want to hand it over to my colleagues who are going to show you how they applied it to their scientific work. So to get us started, I'm very excited to introduce Ana Maria. She is a postdoctoral researcher at La Universidad del Valle in Colombia. And here she works in the Ecology Coral Reef Research Group. And she is going to walk us through the outcomes of applying the approach to one of her recent publications. So Ana Maria, please take it away.
So yes, as Lauren said, I am marine biologist, and I recently published an article about the diversity of microalgae endosymbionts in corals of the genus Poecilopora. This coral is very abundant and is the main reef builder in the Eastern tropical Pacific. So this association between microalgae and corals is mutualistic, and the endosymbiont gives the coral most of its energy to live and grow through photosynthesis. So knowledge about this interaction is fundamental to understand the functioning of this ecosystem. So in this paper, we answer two key research questions. What is the diversity of endosymbionts in the genus Poecilopora of the Colombian tropical Eastern, and is the endosymbiont association is specific to the Poecilopora lineages? So the traditional journal article published online or in the PDF is organized in different sections, like introduction, methodology, etc. In order to identify the main results of the paper and how those results were obtained, it will be necessary to read and review almost all the section of the article. So it works usually. So the relevant findings found in the research are associated with statistical results that frequently can be found in the article text, as we can see there, are linked to a figure that shows those results. So I applied the ORKG Reborn article to the most important findings that answer the key research question that I mentioned before, and I would like to show you how we implement this approach and how it makes my research more useful for you or for other users. So here we can see, as in the same as on a PDF paper, the title, the author, the abstract, that is very similar, but different is that we have the four principal statements that represent the scientific results of the research. So I will focus of two of them to show you how we implement these reborn articles. So in the first results for the first question, the main result was that we found is that endosymbiont community associated with Posilopora corals varied significantly between localities in the Colombian Eastern Pacific. So you can go into each statement and see the details of how the results were obtained, the statistical analysis carried out, the programs, the package, and the data set usage. So you can see here first in the first two colors, you can see the statistical analysis, the variables, and all that is integrated in the reborn article, and also the data set that you use for that. Next, you can have the output. That means you can get the stat table with all the information related with the results. And also you can export it to an Excel file that usually when you see in the PDF paper, you are not able to export it. You have to do it manually. And sometimes that requires a lot of time and effort. For the second statement that I would like to mention is that we found a high specificity of some of the endosymbiont species for the Posilopora lineages. So as I mentioned before, you also can see all the statistical analysis and the programs or all the analysis that underline those results. And also you can have a link for data access. That means that you can download the data and you can have this data to do again the analysis and for yourself. At the end, also you have the statistical table, the figure, and all the implementation that was behind those results. So in this case, we use R software, so you can have the R code to see all the data processing, the analysis, and the graphing that we use to get all these results. So one of the advantages in use the reborn articles is that your research could be increase your visibility and availability of the research findings in different areas of science, not only marine biology. Reproducibility is a fundamental concept, in the scientific research, and it will be used for another users and get more quality of your work. But of course, the reborn articles could be improved if more people are involved and more scientific results are integrated in the ORKG report. So now, thank you.
Thank you so much, Ana Maria, for presenting your work. So the next up, I'm going to introduce Alberto, who recently completed his PhD in the Department of Animal Ecology and Systematics at Justus Liebig University Gießen in Germany. And he's going to walk us through the outcomes of applying this approach to one of his recent publications on a tropical seabird. So take it away, Alberto.
Thank you again, Lauren, for your kind introduction. So I want to share my experience during the 6 months grant with Marcus and Lauren. And yeah, I would like to start my part of this presentation with this, I would say, like a situation that probably everybody at some point in their career or during research has to deal. So let's say that you are doing a really important research and you are looking for one paper or several of them. So you access to the journal, and sometimes if you are lucky, this paper that is really important for your research is an open access. But sometimes depends of the research institution or like at the university that you are part of, maybe don't have access for that paper that you are looking for. And yeah, unfortunately, you have to pay for that paper. Anyways, so let's say that finally you have access for that paper and you go through all that paper, you read and whatever. But honestly, also at some point, you have some questions in the end of that paper. Maybe there is something that you want to get more about a table or a figure. So what I try to share with you is like, in my case, for example, at the beginning of my investigation during my PhD, I saw many of these figures or images that, I mean, I have like a rough or like a brief idea about what is going on, but I didn't know at the beginning what are all the steps that are behind this figure. So yeah, I mean, somebody can say, yeah, you can check in the supporting information or you can check in the methods, but sometimes I will say the length of the papers don't allow us to share everything in the method. So it's really brief, and yeah, sometimes you have to contact the authors, but this is at some point really complicated, the communication. So for that reason, I feel that the reborn articles approach is an option. Basically, this approach transforms the scientific knowledge, as already Lauren said, into a machine-readable format, making that papers more reproducible and accessible across users and, of course, across several disciplines. So now I want to start, I mean, with one of the papers that I have the opportunity to upload and work during this grant. And basically was one of my papers during my PhD that was related with the geographical body size variation in a tropical seabird, the red-billed. And here we work only in the Mexican Pacific because the species breeds in several islands across the Mexican Pacific. And here, basically, the questions that we answered was if the red bill presents a body size across these colonies or islands. Also, if the species exhibit sexual size dimorphisms, and finally, what environmental factors might contribute to these variations in its body size. Here, just because I don't want to bore you, I want to focus just in the first two questions. And yeah, as Ana Maria already also present to you, we are really familiar with this standard format for several editor houses, either Springer, Wiley, Elsevier, that, yeah, on the top, you can see the journal where the paper was published, the title, the authors, and yeah, you have here like an index that you can access for all, directly for all the sections of that paper. And yeah, as well, we have like this, I would say like a standard PDF format or like the PDF paper that you can check with the figures, you can check in the supplementary information. On this case, as I already mentioned to you, I want to focus in the main questions or in the two main questions of my paper. And here, basically, you can see some body traits, the culmen length, the ulna length, and the body mass. So yeah, as I mentioned before, we found like this body size variation across this latitudinal gradient. So that means that the individuals breeding in northern colonies present larger body size traits than those individuals in the south. So you can see how it's like the decrease on these body traits through from north to south. Also, I want to emphasize that in some of the northern colonies, we found these sexual size dimorphisms between females and males just for the culmen and for the ulna. In all cases, males were larger than females. And to get these results, first we had to make some preliminary analysis. And yeah, so basically now I want to jump to the reborn article approach. So also, Ana Maria already mentioned all these characteristics or features that are really helpful or friendly to the users. So I want to jump just quick to the, uh, just give me a second. Here to the main page of the reborn. So yeah, let's say that I'm looking for a specific paper. So I will put here like a body size variation. That is the name of my paper. And yeah, you can click on it. And yeah, as already Ana Maria mentioned, here you can see the title, the authors. You have like an abstract about the paper. And also something that is really nice is this paper in the Reborn platform has its own DOI. So what does that mean? That basically if you get access and you work with the data of that papers is cited, so you can receive a cite from the paper and also from the platform. So I think this is also really useful and helpful for researchers in academia because you have like a wider vision from several, I would say like I would say for several uh, point of views or for several resources. And yeah, also something that is really nice is that, let me back again to the presentation. What's going on here? Yeah, perfect. So yeah, so here, let's say that you read at all the paper and you identify these main results. On this case, here, the body size and the body mass increase from north in females and males. And yeah, the post hoc test revealed that a significant difference between sexes in northern colonies of San Pedro and Farallon de San Ignacio. So here as well, you can check for the statements. So the same, like you can search about these main results by statements in the Reborn platform. And here you have like the culmen length in the red bills shows a decreasing trend from north to south across breeding colonies in the Mexican Pacific. So males from San Pedro, Martir, and Farallon exhibit longer culmen length comparing to females. So you can click in the statement and voila, you have everything here. I would say, in this case, what kind of tests does the researchers or like the authors use? What kind of packages, either like a Python or R, the authors use? Of course, the target in this case was the Coleman length. And also something that is really nice and is really useful is that you have all the input data. What does that mean? That you can have access for the whole data set that the authors use to carry out or perform their analysis. And yeah, you can export to Excel and you can work on it because honestly, you can have the figure or you can check the figure, the tables, but sometimes you want to really get into some specific data that the authors, I mean, again, sometimes it's impossible to share everything in the PDF paper. And yeah, here you have the output data that also in this case depends of the authors. You can have it either on a table format or in a figure format. As you can see on this figure, that also is brand new, created by the reborn approach, is quite similar than this figure that is presented in the PDF paper. And finally, something that is really nice as well is the implementation. So you can have full access for the R script, for the code that the authors use to perform their analysis. And yeah, from my side is everything, and I back the microphones to Lauren. So thank you very much for your time.
Okay, so yes, thank you again, Ana Maria and Alberto, for walking us through how you applied this approach to your work. And now that we've seen a couple of examples, I want to just briefly describe how this actually works. So how do we achieve this? So you understand how you can get involved and how you can apply it to your research. So the way that we do this is that we extend the R or the Python scripts that researchers create originally for their data analyses. So as an example, I'm showing you here a snippet of code from one of Ana Maria's analyses where she was running a Permanova test in R using this Adonis function. And what we want to do now is capture the output of this analysis in a machine readable format. And so we do this using a new R package that we created called dtreg. And we just add some additional lines of code that are going to call a template or a schema that is going to guide us through capturing and structuring key elements of the analysis. So here we happen to be calling this statistical test of difference template that helps us to specify important information related to the input data and different variables associated with this Permanova analysis. So I'm only showing you a subsection of the template. So Ana Maria actually filled out more information than this, but this illustrates how we use these templates or these schema to structure key information from the data analysis. And then finally, we save this schema as a JSON file, which just stores this information in a format that is machine readable. Interlinking this JSON file with the DOI of Ana Maria's journal article is what then enables ORKG Reborn to automatically collect this information via a DOI lookup function. And so this is exactly the work that Ana Maria and Alberto have been doing with their papers and others to produce the Reborn articles that they were just presenting today. We also have a forthcoming package called MWrap that is based on dtreg that will simplify this process of producing machine readable results by packing all of this code into just a couple of lines. So this new package will be available in CRAN soon, so keep your eyes open. And in the meantime, how can you get involved and use the approach yourself? Well, there's a couple of ways that you can help us. So as I've mentioned, this is a novel approach, and we're just at the stage of prototyping this across different research communities. To really get it established, we have set an ambitious goal to create 100 Reborn articles by the end of the year. So the first way that you can get involved is to help us to reach this goal by sharing your data and your code, and then we can implement the approach for you. So just point us to the scientific results that you would like us to capture, and then we can do the rest. So you can send this information, your data and your code, to this email address here, reborn@orkg.org. You can also go to the website, and we have a submission page that restates everything I just said about what you need to submit, and provides the email address there as well. And secondly, our vision really is that ultimately you as researchers will implement this approach yourselves, just as Ana Maria and Alberto have done. And really, thanks to their feedback, we're working to make this process easier, but we still have some work ahead of us. So if you enjoy playing with code, it would be awesome if you could try implementing this approach yourself using the DT-Reg package that I just described. There's documentation that provides vignettes that demonstrate how to apply the schema to different types of data analyses. And really hearing any of the pain points from you, and also hopefully things that work well, can help us to further improve this approach. And our goal really is to design this approach in a way that's useful for researchers. So that means we really need to get feedback early on in this process. It also means that you all have the opportunity to be pioneers of this approach and to shape a tool that could transform the way that we communicate scientific results. We also do a lot of outreach around Reborn articles, so this could be another great way for you to boost the visibility of your work. So I'm going to leave it there for now. I want to say thank you all so much for being at this webinar. This wouldn't have happened without all of you attending. And I especially want to thank again Ana Maria and Alberto for all of the work they have been doing with us the past 6 months, and also for the presentations today and sharing all of that hard work with us.